Okay, so this next video is all about bulk transport. Bulk transport covers two key types of transport that you may have heard of from high school. One of them is exocytosis and one of them is endocytosis. So first off, we're just going to have a quick look at exocytosis. Now, lots of cells do this, so I'm just going to give one example. But obviously, when you're studying this, you need to think about other things that it could be, other types of example. So what this could be a stomach cell, and the thing it wants to release could be enzymes. So think the red blob that I have on here could be my enzymes. Enzymes are made of protein. Proteins are made by ribosomes. So in this cell, there would have to be lots of ribosomes. There would also have to be lots of smooth endoplasmic reticulum, and also a Golgi body or Golgi apparatus in order to package up that protein. And when it's packaged up into a vesicle or a vesicle, you end up with what I have here, the red blob, which is my enzymes, surrounded by a very pale blue vesicle or vesicle. So what is going to happen is the vesicle is going to begin to move towards the plasma membrane or the cell surface membrane. And as it moves there, it'll become part of that membrane. It'll first of all fuse. Because they're both made of phospholipids, they can kind of intermingle and join up. And then it'll become part of that cell, meaning that the contents is in a way like flicked out. Don't write that, but think of it as like flicking out the contents of that vesicle. And what you then end up with is, in my case, the enzymes on the outside of the cell. And that can keep happening over and over and over. You can continually do exocytosis in this way. Now, like I said, there are lots of different examples. Certain white blood cells can produce things like antibodies, and antibodies can be released in the same way. So to summarize exocytosis, first of all, the membrane-bound vesicle will move towards the cell surface membrane or the plasma membrane. It will then fuse with that membrane. It'll become part of that membrane, and then the contents from inside will be released to the outside of the cell. Endocytosis is quite similar, but just in reverse. Again, there are lots of examples of this. I'm just going to use one, which is a white blood cell known as a neutrophil. The neutrophil's job is to go around and eat foreign objects. So I'm going to say, in my example, that the red blob this time could be a bacteria, and the blue cell is a neutrophil. So what can happen? The white blood cell can recognize the bacteria. It can begin to fold around and bend around and move around that object and engulf it. Now, to do that, it has to have recognized it. So it's using things like glycoproteins on the surface. It's got receptors on there to detect what it is. But when it's happy that it wants to take up this bacteria, it will move around the object and it will engulf it. It will then fold on itself, and what you'll end up with is a vesicle on the inside of that cell, or a vesicle. So again, in summary, endocytosis is where a particle such as a bacteria approaches the cell. The particle has key little structures on the surface called glycoproteins. Those glycoproteins are detected by the receptors on the cell surface membrane or the plasma membrane. Then the membrane begins to fold around that particle or fold around that bacteria and enclose the particle. And you then end up with a vesicle on the inside. Now, the example I used was a neutrophil. That's a type of white blood cell. And the vesicle it forms has a very important name that we need to learn called a phagosome. It's just the name given to a vesicle inside a neutrophil. Now, quite an important thing is that both these processes, exocytosis and endocytosis, both require ATP. ATP is hydrolyzed to release energy, and the energy is then used to drive this process, to move these vesicles within the cell, and to either release the contents or to take up the contents. I hope that's been some use. Please look at the key terms, and I'll see you soon.